Booster brings together leading research institutes, universities, and biotech companies to unlock how crops respond to drought and turn this knowledge into practical genetic and biostimulant solutions for more resilient farming. University of Cape Town conducts greenhouse drought stress experiments to produce plant material for genetic analysis from Eragrostis nindensis, also known as lovegrass, a naturally drought-tolerant grass related to teff. The university identifies and transfers the most promising drought resilience traits from lovegrass into teff, studies how the plant recovers after drought, and supports biostimulant development by selecting beneficial microbes from soils where lovegrass grows naturally. Ethiopian Institute of Agricultural Research provides teff germplasm and develops and supplies new teff hybrid varieties for drought-resilient studies. The institute also collects and characterizes beneficial microbes from teff soils and validates seaweed extracts and project-develop microbial biostimulants through greenhouse and field trials under Ethiopian drought conditions. University of Bern examines how different teff varieties respond to drought under controlled conditions, working closely with EIAR. By testing seaweed extracts and beneficial microbes, the team identifies the most effective drought resilience traits, studies how plants cope with water stress, and shares these findings with project partners to support the development of more drought-resilient maize. Heinrich Heine, University of Düsseldorf conducts greenhouse drought stress experiments on maize using both public European and industry germplasm and applies molecular and computational analyses to identify key genetic control points linked to drought resilience. By comparing genetic responses across maize, teff, and lovegrass, UDUS helps reveal shared drought response patterns and supports the transfer of resilient traits across crops. Council for Agricultural Research and Economics, the project coordinator, studies how teff and lovegrass respond to drought using plant materials provided by project partners. CREA integrates and compares genomic data across crops, validates maize drought responses using advanced plant model analysis, and contributes to maize field trials in Italy's Po Plain to test newly developed biostimulants under real farming conditions. Flemish Institute for Biotechnology uses precision genome editing to make small, targeted changes to maize genes, helping plants perform better when water is scarce, and tests these genome-edited plants under greenhouse drought conditions. The institute also transfers promising traits from teff and drought-tolerant lovegrass to maize, selects microbial candidates from maize soils, and studies how the most effective microbial solutions work inside the plant with project partners. BioAtlantis develops tailored seaweed-based biostimulants designed to improve crop tolerance to drought. The company leads the formulation, greenhouse screening, and scaling of seaweed extracts for maize and teff, and coordinates field trial design and data analysis to assess the performance of seaweed-based and microbial biostimulants under real farming conditions. Aphia.bio develops microbial biostimulants to improve crop resilience to drought. Starting from beneficial microbes collected from maize soils, the company screens and scales the most effective microbial candidates, formulates them into seed coatings, and leads maize field trials to test them alongside seaweed extracts, assessing their performance under drought conditions in southern Spain and Hungary. Center of Plant Systems Biology and Biotechnology tests seaweed extracts on maize under greenhouse drought conditions to identify the most effective treatments. The center studies how these extracts work inside the plant by analyzing metabolic and biological responses, and leads maize field trials in Bulgaria to validate their performance under real farming conditions. From African field trials to European laboratories and farms, booster partners work together to transform drought science into real solutions that strengthen crops, support farmers, and safeguard future food security.